Thanks, Cord.
Thanks very much, Mark. This presentation, ladies and gentlemen, is supposed to answer the question why leptospirosis continues to occur in workers of vaccinated dairy herds. And I present this on behalf of PhD students Uni Jupiana and my colleagues in the lepto team at Mesa University, Emily Vallée, Jenny Weston, Peter Wilson, Julie Collins Emerson, Jackie Benchop. By way of introduction, the incidence of notified human leptospirosis in New Zealand is the highest among industrialized countries. People get infected by droplets of infected urine that carry the bacteria through eyes, nose, mouth, or skin lesions. Leptospira colonizes the kidneys, and infection can lead to very severe flu-like illness. Thank you. Livestock and wild animals are the main infection sources. They contaminate the environment and expose farm and forestry workers. Dairy workers are highly exposed through exposure to urine twice a day while milking. There are 7 serovars in New Zealand, of which Hardiobovis pulmona are the most widely distributed and therefore included in vaccines. Others are Copenhageni, also in some vaccine types, and then the non-vaccine serovars, Tarasovae ballum, and very rarely found, Canicola australis. The adoption of vaccination in the 1980s is thought to have reduced notified cases from over 600 in the 1970s to about 100 per annum since 1997. More than half of the notified cases are from farm workers. But despite about 95% of dairy farmers using vaccination, dairy farmers are still appearing among the notified cases. Therefore, a fairly recently pilot study found that 1 out of 3 herds and 3 vaccinated cows had positive urine PCRs, suggesting continued shedding. Unfortunately, at the time, no blood was collected at that study, so we couldn't relate the shedding to serovar information. I was a chairman, I don't know how to operate this thing here. Therefore, we implemented this study. It was a cross-sectional survey, a random selection of 200 farms from the National Dairy NZ database, where sampling was done in January to March 2016. These herds were from all regions of New Zealand and were further stratified by herd size, and you can see from the circles that the larger the circle, the higher the sample, and small areas with a small dairy population had a few herds included, like the West Coast here, but the big areas, Southland and Canterbury, Waikato, buried the load of the sample. Within the herd, then 20 cows were sort of conveniently randomly selected and bled, so blood was collected and urine was collected, and serology was done by the macroscopic acclination test at a cutoff of 1 to 48, and urine was PCR tested with a Gyrase B primer. You see the results here, herd level results, about 1 out of 4, or 27% of the herds shedding. You can see the distribution of that over New Zealand with slightly a gradient from north to south. More herds were positive in the north and slightly less in the south, possibly related due to warmer climate in the north and more rainfall. At cow level, we had about 2.4%, slightly less than the pilot study, were positive, PCR positive, suggesting that they shed live leptospira. Now then the serovar information, firstly, not surprisingly, we had positive titers for vaccinated dairy cows and vaccine serovars that were reasonably high. Well, this was about 7 to 8 months after sampling, so the titers decay rather quickly, so therefore you have less than half were seropositive for Hardiobovis and even less than for Pulmona Copenhageni, and that about reflects the decay of the titer duration. More importantly and more interestingly are the non-vaccine serovars, where clearly Tarasova was standing out, and 3 out of 4 herds, 70% of the cows, whereas Copenhagenite Ballum were at a low rate. Ballum usually been associated with pigs, and Tarasova traditionally be associated Ballum associated with rats, and Tarasova associated with pigs. Now, the next question then was to associate these serology results with shedding, and that's what you see here. 4 out of these 5 serovars had no relationship whatsoever with shedding, which is sort of a good news in terms of saying that vaccination seems to be successful, because these vaccine serovars were not related to shedding. But on the other hand, Tarasova was standing out, coming up with a very, very strong association, explaining almost all of the shedding in the sample. There was a nice dose-response effect. It was almost too nice and clear to believe. We rarely get these clear results from observational studies. And it also shows that the higher the Tarasova titer, the higher the probability of shedding. That means the bug successfully evades the humoral immune response and successfully hides in kidney tubules. It's an intracellular bacteria. So that might seem trivial to you. For us, it rocked the boat. That was really surprising. So we became interested and said, well, what has happened? What's the problem here? We wouldn't have expected this. What kind of bug is this? So we sequenced 75 of these amplicons, PCR amplicons, out of the 94 positives that we had, and tried to relate them to known sequences from those primer PCRs, and found that about, I think it was 52 of them, were not really very genetically related to this group here, Hardy bovis. Well, a few were Tarasova or Balkanica or Ballum. This is the typical Bok-Petersenia group. But there was 51 of them that were standing out, and we couldn't relate them really, and called them species X, or Tarasova type X. So then the question was, what happened? What have we missed here? And we looked back into all the studies that had been done previously since 1953, when Kirschner did his first 100 dairy cows and tested them, and found a few of them, 3%, positive for Tarasova. And then there was a number of studies that didn't find any. Large studies here, 890 cows being bled and tested for Tarasova, 0 positive. Then some others, and the maximum that was found here in 1976 was 6%, up to 20% of the herds. But that depended on how many cows in a herd were sampled. And then 35 years, nothing happened. Everybody sort of was happy that the rates of dairy workers getting ill was going down, and the researchers were riding to glory on a downward slope of an epidemic curve, and nobody had ever been looked into dairy cows anymore until the pilot study happened. And then the survey came up, and there we are. We've got 74% herds being positive, and 17% of the cows being seropositive. So what do we do with this? Well, what reasons might occur, might be associated with that, or have caused this increase of this emerging new strain? Well, you think of things like increasing herd size, more intensive production, economy of scale, increased antimicrobial use, or climate change. All of those things might be responsible, but we clearly missed the boat on this case. So another question is, of course, then, well, does this new strain cause disease? And there's a study published in 2012 where it was suddenly found out that 1 out of 3 of those cases were dairy workers. And not only that, these dairy workers were 7.5 times as likely to test for Tarasova compared to other workers. So clearly there was disease in dairy workers caused by Tarasova. And then digging deeper into more cases, 1,500 cases over a longer period of time, again, those that were caused by Tarasova, a large proportion of them were dairy workers. Another clear incidence that this new serovar actually caused dairy workers to fall ill, but we've never noticed. Why not? Because the surveillance system only showed farmers in their statistics. They didn't differentiate what kind of farmers they were. So in conclusion, the number one and the good news really is that vaccination against Hardiobovis pomona, and well, a few of them used Copenhageni as well, was apparently effective because there was no relationship with shedding. On the other hand, a small percentage, well, 3%, but at least, well, a reasonable large proportion of herds continued to shed leptospira and expose dairy workers. Now, Tarasovi, we now know, is the main reason for shedding and for disease in dairy workers. And I could tell you stories about herds not using any vaccine, what that means for how high the risk is of having dairy workers falling ill with this really terrible disease. 75% of the herds have evidence of Tarasovi, and if we would have bled more than 20 cows per herd, this would probably increase further. But more than half of these shedders are what we don't know what it is really, is Agent X. And so we now start trying to grow this bug. We clearly have an emergence of a new strain. Does it also find a reservoir in cattle? Well, there are arguments in favor of that. Because Tarasovi does not cause illness in cattle, not that we knew it or that we've been asking for it, but farmers haven't observed any signs. No abortion, nothing happening to these cows. So that's number one, and it occurs in dairy cattle as much as in beef cattle as well. It seems to be maintained in the population, well, in the system of dairy cows in their environment. And not being dependent on external infection or addition from an external source. So the criteria for a reservoir for this new strain. Now, of course, dairy vaccine companies want to add Tarasovi to their vaccine. That requires that we find it and characterize it very carefully and compare it to any laboratory strains or vaccine plant strains that are available, because we want to make sure that we get the right strain into the vaccine that also triggers an antibody response against the correct one. And that's the question at the moment. We'll try to culture it, and we can't. I haven't found a positive culture yet, but even though the one that we have for our MAT test is growing very happily. So there's something new with this, totally new about this new strain, and that is ongoing work at the moment. So that's my message to you and the answer to the question, and I thank you very much for your attention.